the opportunity to visit, come here and participate in the memorable event. And my association with Gobind and Tom goes way back to 1972. This is the group picture taken in 1972 at MIT. And Tom is here, and Gobind, and I'm standing here, and Revu is here, and Gobind, and then it's myself and Marvin Crothers, and Rama who is here, and I don't see very many people in this picture at the audience.
Can I just point to one person?
Yeah.
The fellow back row. This fellow here with the hair.
Yeah.
That's my dad.
That's a yeah. So the following year, Gobind came to Cornell to work with Ephraim Racker, and he was assigned only one small bench here in his lab. In his lab, he had a small bench. He worked very hard in the lab, and this is the occasion of 75th birthday symposium for Leon Heppel, who is a legend in amino acid transport in E. coli. At that time, all these luminaries, Dr. Kornberg and Ephraim Racker and Gobind Khurana and Revu in this picture. So it was a wonderful experience. In Gobind's lab, I really learned quite a lot how to synthesize oligonucleotide, which resulted in 3 publications. But we spent like 6 months to synthesize 3 oligos. So now we are working with the dengue virus, and my collaborator in this project is Choi, Kuyung Choi. She is a faculty member at the Department of Biochemistry and Molecular Biology at the University of Texas. And so dengue belongs to Flavivirus genus in the Flaviviridae family, and there are 4 serotypes of dengue. And this Flavivirus family consists of more than 70 human pathogens. They are mostly transmitted by mosquitoes or ticks. The genetic relationship in this phylogram is shown here, and you can see that dengue 1 to 4, they are closely related, and then the rapidly spreading Zika virus is more closer to dengue, and the Japanese encephalitis and other encephalitic flaviviruses are a little distant from this, and the prototypic member of this flavivirus is yellow fever virus. So dengue is caused, the disease caused by dengue is by infection with any one of the 4 serotypes. Dengue is endemic in more than 100 countries, including India. The estimated frequency of dengue virus infections has been revised, is 390 million people annually, and most of them, of course, is asymptomatic. The person infected with dengue, they don't even know that they were infected with mosquito-carrying dengue. We sequenced the dengue genome, New Guinea C strain, which is a prototypic strain of dengue type 2, manually using Maxam and Gilbert and Sanger dideoxy sequencing. Dengue virus is the enveloped virus. It has a positive strand RNA encapsulated inside with capsid protein, which is a very basic protein, and the double lipid membrane. And then on the outside is envelope and pre-M protein, which are the targets for immune response. And this organization, this envelope virus is similar to other envelope viruses also. So this is the life cycle of the dengue virus. The virus binds to specific receptor and is internalized and goes to the endosome. The acidic pH causes the conformational change of dimeric envelope to trimeric envelope. And then there is a release of viral RNA, which is translated in the endoplasmic reticulum by the host translation system. It's a cap-dependent translation. And then the polyprotein processing by the viral as well as cellular proteases take place. And then it goes to the mature proteins assembled into replication complex, and viral RNA is replicated in membrane compartments of endoplasmic reticulum, and then the immature particles containing precursor membrane and the envelope protein form a heterodimer and encapsulate the viral RNA complex with capsid and goes to the Golgi, where the maturation takes place, and this again becomes that envelope becomes a dimer and is released into the circulation. So the dengue RNA is a single-stranded positive strand RNA. It's cap-dependent. Cap is at the 5' end. There is a short 5' untranslated region and one long open reading frame, and then there is a 3' untranslated region. These two are highly structured, and these are important for both translation as well as replication. This is the topology of polyprotein in the ER membrane. This is the cytoplasmic side and then luminance side, capsid and precursor membrane, envelope, and NS1, they are on the luminance side. And then there are integral membrane protein, viral proteins. And then this two cytoplasmic protein is NS3 and NS5. So these two proteins are very important for viral replication. So we made antibodies against these two proteins, and then we showed that these two proteins exist as a complex in the infected cells. Using one antibody, you can bring down both proteins. So at that time, we didn't know what was the enzymatic activity, what were the enzymatic activities of these two proteins. But when those enzymatic activities became known, then the function was now could be analyzed and then tested and analyzed. And so one of the NS3 protein is a multifunctional protein. The N-terminal part of NS3 has NS3 is a protease domain, serine protease catalytic domain, but then it requires NS2B, which is the integral membrane protein in the ER, for interaction and activation of the protease domain. The C-terminal part of NS3 has ATP-dependent RNA helicase activity, and NS3 also has a 5' RNA triphosphatase activity, removal of the gamma phosphate to form a diphosphorylated RNA, which is, this first step is important for capping at the 5' end. The functional, NS5 is also a multifunctional protein. The N-terminal part of NS5 has conserved residues in the methyltransferase, and the C-terminal part has RNA-dependent RNA polymerase. This activities were later on established, RNA methyltransferase by Piang Shi's lab, and then we were working on the RNA-dependent RNA polymerase. We showed that this E. coli express polymerase can initiate de novo the minus strand synthesis from the template RNA. So this is to study the replication of a mechanism of viral replication of RNA using in vitro assays. First one to establish an in vitro system using membrane-bound replicase complex. So another flavivirus, Kunjin virus, was used to infect mammalian cells, Vero as well as BHK. Westerwey in Australia showed that one can isolate the cytoplasmic membrane fraction and show that this has an activity to synthesize RNA as long as you provide the 4 triphosphates, one of which is labeled. And recently we have now purified this membrane complex by using differential centrifugation, isolate the microsome, and we showed that it's active in RNA synthesis. And we also are following this proteomics of this microsome to see what all the proteins associated with this. So this is the Westerwey system. If you take the cytoplasmic fraction and then add the labeled triphosphates, in a time-dependent manner, you can show that there are 3 species of RNA. The slowest migrating species is the replicative intermediate, and then intermediate mobility. The virion RNA is single-stranded RNA, which is the progeny RNA. And then this faster migrating replicative form is a double-stranded RNA form. This is completely resistant to RNase, and this is completely sensitive to RNase, and this is partially sensitive to RNase. Based on this, Westerwey proposed this model that positive strand RNA is copied into form the double-stranded replicative form, and then there is unwinding of the double-stranded RNA at the end, 3' end of the negative strand, to synthesize a new positive strand, and displaces the parental strand, and then it is capped at the after the displacement. So this is not clear yet whether capping occurs co-transcriptionally or it occurs post-transcriptionally. So we have submitted a grant application to analyze this aspect. And so the capping requires 4 enzymatic activities. First step is again dephosphorylation of the triphosphorylated RNA, and then addition of GMP after hydrolysis of GTP by NS5 methyltransferase, and then capped RNA is then methylated by again NS5 methyltransferase, and it puts a methyl group at the 7 position as well as 2' O methyl position. So recently, my collaborator Kyung Choi solved the structure of full-length NS5, dengue NS5, and she showed that it's a dimer. And then she proposed the model that if NS5 is involved in both RNA synthesis as well as capping, the methyltransferase domain is in the N-terminus, and the RNA-dependent RNA polymerase activities in the C-terminus of NS5. And if that protein is involved, there will be a problem with a monomeric NS5 because when the template, positive strand RNA template enters here, and then double-stranded RNA exits here, and then the methyltransferase domain is farther away on the other end, so there is a need for a large conformational change. So this is, so she proposed that this is, now she knows that this is a dimer, so she proposed that maybe a dimeric NS5 is involved in capping. And So this is a model she proposed and published in this paper in PLOS Pathogen. So we were studying in a different, by a different approach. Since NS5 has a distinct function in the virus life cycle, 5' capping as well as RNA synthesis, we asked if chimeric NS5 containing these domains from different dengue virus serotypes, can it function well in viral replication? Because even when, even though these virus serotypes are evolved from a common ancestor, but because these proteins are designed to function in replication of that particular RNA, the serotype RNA, can you mix match them? So we asked this question by putting the methyltransferase domain from another serotype, dengue type 4, and then made a chimeric RNA and see the replication of these serotype. This is the sequence alignment of the dengue type 2 and type 4. It's pretty homologous. I mean, 72.6%. The full-length NS5 sequence identity is 74%. And when we did that, we found that the chimeric NS5 replicates very slowly. I mean, in 10 days, you don't see any positive replication. And 22 days, you start seeing some replication. And 30 days, you see more. And so we repeated this experiment 3 times. Twice, we got this K74 isoleucine, and in third case, there was a mutation in the NS3. This K74I was in the methyltransferase domain of dengue type 4, and this is mutation occurred in the NS3 RNA helicase so there is an interaction communication between methyltransferase domain and NS3 during replication. And we also quantitated the viral RNA by qRT-PCR, and we showed that at 30-day supernatant had almost restored the RNA copy number almost to the wild type level. And the one thing surprising is there is no difference in the enzymatic activity of wild type versus chimeric NS5 or even with adaptive mutation. And also no difference in the capping activity of these NS5 molecules. Then we switch the other domain, the C-terminal domain of dengue 4 polymerase, and then the N-terminal part is dengue 2, and the whole backbone is dengue type 2. So we asked whether this will replicate or not, because polymerase domain is also highly conserved between dengue type 4 and type 2. And again, the answer is same. The polymerase chimeric virus is severely attenuated, and it takes a long time, 30 days, to regain replication fitness. And then we sequence these by using Sanger sequencing methods. There was only single, in the first case I mentioned to you that K74I was the single mutation identified by Sanger dideoxy sequencing method. And the second, one of the third experiment, we saw that there is a D290N, whereas in the case of polymerase, again, K76I was generated, and then in the second experiment, there's E706 to aspartic acid was generated. So these adaptive mutations restored the replication fitness of the chimeric virus. So we have also analyzed the sequence analysis by RNA-seq, and and identify, this confirmed some of these mutations. Additional mutations also revealed by RNA-seq analysis. And when we map these mutants on the crystal structure of NS5, we found that the residues, they were mutated, they all line up on the NS5 monomer-monomer interface. So this suggests that, again, suggests that NS5 may function as a dimer. So flavivirus NS3 and NS5 play very key roles in viral replication. These proteins are endowed with multiple enzyme activities, which are important for viral replication and 5' capping, and hence these are targets for antiviral drug development. And the flavivirus replication complex is assembled in the ER and consists of viral non-structural proteins, unknown host proteins. The serotype specific protein-protein interaction and protein-RNA interactions are likely important for viral replication. Our results indicate that NS5 dimer is the functional form in replication. So we have to do further work. We have submitted a grant application. We don't know what's the fate of that now. So I end my talk now, I think. Thank you very much. Thank you.
Any questions? Here, let me give you this microphone.
Sir, as you have already told that adaptive mutation is a kind of action going on there. Adaptive mutation. They are reverting back to the replication form. They are reverting back to the replication form after few days in the chimeric forms. So that reversion is because of those adaptive mutations. It's going on.
This all we have done so far. So we haven't isolated the replication complex from the anti-infected cells. Especially initially, the chimera RNA transfected cells takes a long time to replicate, right?
So that is, you have concluded that they are going under some adaptive mutation because of which they are reverting back to the replication form after few days. That you have concluded, no? I will discuss it.
Yes. Let me see if I can just rephrase the question as I heard it. Something like the D190N mutant, is that now more replication competent? Is that what you were asking in some ways?
Whatever chimeric gene combination you were using, the methylase and polymerase chimeras, after a few days they were reverting back to the replication form. That is because of those adaptive mutations.
Adaptive mutation is the
Adaptive mutation is basically a problem with the virus.
yes,
Reverting back.
but yes, right. And you want to know whether we have isolated the.
Basically, my question is that adaptive mutations are always conserved or they are different? Whenever they are getting something new, they are mutating, those are at the same specific codon or it's a different every time? They will different?
I suspect we'll continue this particular line of questioning at the break. Are there
Actually,
you have an answer?
We don't have an answer because these are all the work we have done. We have isolated the replication complex from the chimera virus RNA with the adaptive mutations. Those are all the work to be done in future. Yeah, that's it.
Additional questions, Roger?
Quick question, quick answer. Why it is so hard to make vaccine against? Why is it so hard to make a vaccine?
Oh, yes.
quick
Yes.
answers. Go ahead.
Okay. The reason is there are 4 different serotypes. There are 4 different serotypes. The vaccine has to be effective against all 4 serotypes. If one of the the vaccine is not efficient in protecting one serotype, as in the case of dengue type 2, the vaccine, the candidate vaccine only protects 60% cases, only 60% protection. So that's not good enough because it can lead to antibody-dependent enhancement. The vaccine, antibody against the vaccine could form an immune complex with the secondary infecting virus, and it can be taken up much more efficiently by any cell which expresses Fc gamma 2A receptor. So this way the viral load becomes very high, and the vaccine cannot really handle that. So the only way FDA is going to approve is a tetravalent vaccine. So that has been a challenge. I think Sanofi has been working on this for a number of years, and I think they have a candidate vaccine which is now approved in other countries like Mexico and other. So we have to see what's the results of those.
Yes, we have time for one more question, actually. Question over here. Where? Here.
So this NS5 is important for replication of the virus. So is it possible to target, because these days antibodies are being used as a drug as a drug itself. So can we generate some antibody against this NS5 and use it as a drug and target this NS5 as a drug target for the virus?
Yes, it is possible. Yeah, I don't think anybody is working on that actually. Antibody therapy for
the dengue virus.
Dengue virus.
But it is, since you have shown that it is important for replication,
Yeah.
so it could be something that can be tried.
NS5 is a polymerase. Is an excellent target for antiviral drug development. In fact, hepatitis C, the currently marketed drug is actually one of the candidate drugs is against HCV polymerase. And also the other one is protease. So it's a combination
So if, as you are saying that there is a drug that is being used in hepatitis, so is it possible to reposition that drug and test it in the virus also?
Yeah, it's possible.
Has it been done?
No, I don't think has been done. No, the focus is on Zika virus because Zika caused so much havoc in the South America, North America, and even southern part of North America. So now the focus is, all the funding goes to Zika. So I don't know whether dengue is not a problem in US. Unless somebody like World Health Organization or Gates Foundation funds this research, we don't know.
Well, thank you very much. Yes. So our last speaker in this session is Joydeep Das, who's over there. And Joydeep did his PhD in Bombay at the Indian Institute of Technology and did postdoctoral work with Gobind and is now a fellow Texan. He's at the University of Houston, where he began as an assistant professor and is now an associate professor. And his interest is in alcohol at the professional level, at least, and particularly mechanisms of addiction. And he's going to tell us about his very beautiful work today in that area. And thank you.
Thank you.
Yes, sir. Please.